Welcome everyone. In this video I will analyze another exam question which focuses on how Cish deficiency affects CD8 positive T cell signaling and function. Let me just remind you first that Cish is known as a negative regulator in cytokine and TCR pathways, and here its role is dissected using knockout mice. Starting with panels A, B and C, we see reporter assays measuring transcriptional activities downstream of key pathways after TCR stimulation by anti-CD3. Look at panel A first. STAT5 activity, which is mostly driven by cytokine signaling, shows no significant difference between cis knockout and wild type CD8 positive T cells. This indicates that Cish deletion does not alter STAT5 signaling downstream of the TCR. Move on to panel B, please. Now this one reveals a clear increase in NFAT transcriptional activity in Cish deficient cells. Look at the white bars, both in magnitude and duration. NFAT is a critical transcription factor activated by TCR signaling and calcium flux. Similarly, panel C shows that NF-kappa B activity, another key TCR-driven pathway involved in TCR activation and survival, is significantly elevated in cis knockout cells, white bars, compared to wild type. Panel D measures phosphorylation of STAT5 after IL-2 stimulation, confirming that cytokine-driven STAT5 activation remains unchanged by Cish loss. As you see, white and black points overlap in all three diagrams, reinforcing that Cish does not regulate the cytokine signaling axis. Finally, panel E shows calcium flux, which is one of the earliest and essential signaling events after TCR engagement. Cish knockout cells exhibit enhanced and prolonged calcium flux, red line, compared to wild type, consistent with increased downstream NFAT and NF-kappa B activation. Together, these data demonstrate that Cish selectively restrains TCR proximal signaling pathways, including calcium signaling, transcription factors NFAT and NF-kappa B, but does not impact cytokine-driven STAT5 signaling. This selective inhibition likely modulates CD8 positive T cell activation and function, underlying the enhanced anti-tumor immunity observed in Cish deficient models.